Okay. Good morning, everybody. Sorry for the inconvenience occurred due to the delay. Let's start the new lecture on nervous tissue. This is regarding your CNS module. Okay. The nervous tissue. It is a tissue just as other tissues, which is designed to deliver rapid and precise communication between different parts of the body by the action of specialized nerve cells called neurons. So, in the other tissue variants in the body, there are specific nerves for the cells which comprise of that particular tissue. Likewise, in the nervous tissue, the specialized cells are called as neurons. So, the the nervous system, we can divide in various ways. Mainly, it can be divided into two main parts. Those are the central nervous system, which is comprised of brain and the spinal cord, and the peripheral nervous system, which is comprised of nerves run between the CNS and other tissues, and the nerve relay stations, which is called as ganglia. Functionally, we can divide the nervous system as the somatic nervous system, which is responsible for the voluntary function in the body, and the autonomic nervous system, which is responsible for the involuntary function. Histologically, there are various cell types in the neurons. So, various cell types in the nervous tissue. Those are basically we can categorize into two as neurons and their supporting tissues. These supporting tissues are called as neuroglia or the glial cells. So, when we talk about the components of the nervous tissue, as histologically, we can divide them into neurons and the neuroglia or the glial cells. So, when we look for the differences and the basic characteristics of these two cell types, the neurons are the primary type of cells that associate with the nervous system. So, the neurons are the highly specialized cells in the nervous system, which is responsible for propagate the signals. So, these are highly specialized and they are interconnected with each other. That's how they are able to transmit the signals, and they gather and process information and then generate appropriate response signals. And the neuroglia or the supporting cells, they are the cells known to play a supporting role for the nervous tissue. So they are the supporting tissues, supporting cells of the neurons. They give both mechanical and the metabolic support. And it also gives defense and immunological function. They are the immunological cells in the nervous system. And also in the peripheral nerves, they provide sheath. They provide sheath to the peripheral nerves. And in the brain, they line the brain cavities. They act as the epithelial cells of the brain cavities. And there is a small special thing in the brain and the blood vessels, between the brain and the blood vessels, which is called as the blood-brain barrier. These neuroglia cells are participating in forming the so-called blood-brain barrier. And this is the structure of a neuron. I think you have studied this in the foundation course also. The neuron, the shape of the neuron is very specific. There is a large cell body. In that, we can see the central nucleus and the cytoplasm. And also, there are small processes extending from the cell body, which are called as the dendrites. And there is one or more long and large process. It is called the axon. And this axon, the end of the axon is divided into small processes. The end of these small processes are called as the axon terminal. And at the end of the axon terminal, we can see the synaptic end bulb. The place where the axon starts from the cell body is called as the axon hillock, which is a slightly expanded area. And along the axon, we can see myelin sheaths. These myelin sheaths are formed by the neuroglial cell called Schwann cell. This table shows what are the structures of neuron cell and what are their functions. So the main part is the nucleus. As in the all cells, the nucleus functions as the control center of the cell. It contains the DNA. Then there is the plasma membrane. Plasma membrane covers the cell. It is responsible for the reception and transmission of impulses, and it has the facility of selective permeability. Covered by the plasma membrane is cytoplasm. Cytoplasm contains various organelles and inclusions. There is a special granules called micelle granules in the nervous cells, neuron cells. They are the responsible granules for the protein synthesis inside the neuron. And there are the same organelles which can be seen in an ideal cell, mitochondria, which is responsible for the energy reproduction and cellular respiration, and Golgi apparatus for storage of secretory products, and also neurofibrils, which give the support to the cell body and the stability. And the axon and the dendrites, which are the special features seen in the neurons. So this axon is the process which is responsible for carrying impulses away from the cell body. when a signal comes to a neuron cell, electrical impulse comes to the neuron cell, axon is the one which carry that impulse or the signal away from the cell body. It is the one which gives away the signal to another neuron cell. So it is called as the efferent part of the cell. And the dendrites are the one which receives the signals and carry the impulses into the cell body. So they are called as efferent processes. Okay. So there are few types of basic neuron types. We can divide them into according to the structure. This picture shows those basic neuron types. Okay. This one is the multipolar neuron. This one is a bipolar neuron. And this is a pseudo-unipolar. This is a unipolar. And this one is a apolar neuron. Apolar means there is no pole. That means the all processes are similar. And the unipolar one, it has cell body in one corner and one axon starts from a side of the cell. In the bipolar neuron, there are two axons. There are two main processes. Sorry, not axons, two main processes. One is the axon and the other one is divided into the dendrites. In multipolar neuron, there are several processes starting from the cell body. The large one is the axon and the shorter ones are the dendrites. So this table shows the characteristics of the neurons. Apolar neuron has no process. And unipolar has only one process. Bipolar one, two process. Axon, one axon and the dendrite and one dendrite. They are not common ones. The examples for bipolar neurons are the olfactory nerve cells and the retina and ganglion of the eighth cranial nerve. Multipolar neurons has many processes, one axon and many dendrites. So they have many poles. And also we can divide the neurons according to their function. Efferent or sensory neurons and the efferent or motor neurons. So efferent or the sensory neurons are the nerve fibers which are responsible for bringing sensory information from the outside world into the brain or the spinal cord. So they receive sensory inputs or sensory signals from the outside world. They grasp them and bring them to the brain or the spinal cord. This sensory information involve special sensors such as vision, hearing, touch, temperature, smell, taste, and those things. Different neurons or the motor neurons are the ones who carry signals from the brain to the peripheral nervous system to initiate an action according to the impulses received from the efferent one. So they carry the impulses from brain or the spinal cord to the muscles and the other organs. Then we have these special neuron types in the cerebral cortex. So, these are these cells. Some cells are named according to their shape, some are according to their features, some are according to the person who first described the cell type. Okay. So there are, you can see there are 5 main cell types in the cerebral cortex. These 5 main cell types are arranged according to 6 layers. So the main cell type is the pyramidal cells. They are named according to the shape of the cell bodies. They are cell bodies shaped as a pyramid. Its apex is directed toward the cerebral cortical surface. So, and the base directed toward the inner part. And there is an axon which is started from the base of the cell, and it passes on to the underlying white matter and synapse in the deep layers of the cortex. The apex is the place where a thick branching dendrite starts. So this dendrite passes towards the surface. And also there are short, small dendrites which arise from the edges of the base. So among these pyramidal cells, there are another special cell type which is called as bed cells. These are the huge upper motor neurons of the motor cortex and are the largest of the pyramidal cells in the cortex. So the most largest pyramidal cells are called as the bed cells. The second cell type is the stellate cells or the granular cells. These are small, rather small compared to the pyramidal cells. And they have a short vertical axon and several short branching dendrites. They have a star-shaped cell body. This is a stellate cell. It has a very starry shaped cell body and a long axon and short dendrites. They too have subtypes which are called basket cells and the neurogliaform type of stellate cells. The third cell type is the cells of Martinotti. These are small polygonal cells and they have short dendrites with the axon extends towards the surface and bifurcates to run horizontally. These are the cells of Martinotti. They runs to the most superficial layers. And the next cell type is the fusiform cell. They are spiral shaped. They are spiral shaped and oriented in the right angles to the surface of the cerebral cortex. They have an axon arising from the side of the cell body and it passes to the superficial layers of the cerebral cortex. And there are dendrites from the each end of the cell body. The last cell type of the cerebral cortex is the horizontal cells of Kajar. These are small spindle-shaped cells only found in the most superficial layer of the cerebral cortex. They lie parallel to the surface and least common cell types. Their axon passes laterally. These are the horizontal cells and they are in the most superficial layer. Their axon pass horizontally and synapse with the dendrites of the pyramidal cells. So, the above mentioned cell types are arranged in 6 layers of the cerebral cortex. So, from the very most superficial layer, it is the flexiform or molecular layer. It contains horizontal cells and the neuroglia. And the second layer is the outer granular cell layer. It has small pyramidal cells and the stellate cells. Third layer is the pyramidal cell layer. This layer has moderate size pyramidal cells. And when the pyramidal cell layer goes deeper and deeper, the size of the pyramidal cells get large and dense. Also, there are Martinotti cells. The fourth layer is the inner granular layer. This has densely packed stellate cells. The fifth layer is the ganglionic layer. This ganglionic cell layer has large pyramidal cells. So, it is the place where we can see the bed cells, because bed cells are the most largest form of the pyramidal cells. So we can see bed cells in the ganglionic layer. Apart from that, there are a small number of stellate cells and the cells of Martinotti. The innermost layer is the multiform layer. There are numerous small pyramidal cells, cells of Martinotti, stellate cells, and fusiform cells. So the variety of the cells in the morphological forms are very wide. Similar to the cerebral cortex, there are several neuron types in the cerebellum also. We can see 5 cell types in the cerebellar cortex. Those are the Purkinje cells and the granular cells, stellate cells, basket cells. Those are the things. So the Purkinje cells are the largest cell type we can see in the cerebellum. They have very large cell bodies and extensively branching dendrite system. Their dendrites are extensively branched. Here we can see severely branched Purkinje cells. And stellate cells and the basket cells. They are scattered in the molecular layer. And there are Golgi cells. These are the Golgi cells. Golgi cells are scattered in the superficial part of the granular cell layer of the cerebellum. And there are also some granular cells. So this picture shows the cell layers and the cell types of the cerebellum. So the most superficial layer is the molecular layer. The most inner layer is the granular layer, and in between there are Purkinje cell layer. So in the outer molecular layer, there are few neurons and large number of unmyelinated fibers. The inner granular layer is extremely cellular compared to the outer molecular layer. And the middle layer contains of single layer of the Purkinje cells. It's rather simpler than the cerebral cortex. So far we have talked about the neurons and the various neuron cell types we can see in the cerebral cortex and the cerebellum along with their layers. Now, the other part which comprise of the nervous tissue is what? It is the supporting cells or the glial cells. So, neuroglial or supporting cells, we can see various supporting cells in the nervous system. Some are in the central nervous system and some are can be seen in the peripheral nervous system. So, if we divide them in a basic way, we can see 5 main cell types in the nervous system. 5 main neuroglial cells. Those are neuroglial cells, ependymal cells, capsular or satellite cells, Schwann cells, and other supporting cells. Among them, the neuroglial cells can be further divided into macroglia and the microglia. So, macroglia can again divided into astrocytes, oligodendrocytes, and the glioblasts. The function of these different cell types and the sites we can find them are different. So, astrocytes, again we can divide into protoplasmic astrocytes and the fibrous astrocytes. These protoplasmic astrocytes are found in the gray matter and they are thick and symmetrical. Fibrous astrocytes are found in the white matter. They are thin and irregular. Now, let's see what are the important features and the functions of these various neuroglial cells. This table will show you a brief description and notes on function of these various neuroglial cells. So, mainly the neuroglial cells found in the parenchyma of the brain and the spinal cord. And their main function is to give mechanical and metabolic support to the neurons. And the ependymal cells, these are specialized epithelial cells which line the ventricles and the spinal canal. Ventricles of the brain and the spinal canal is lined by the ependymal cells. So, they have a specific function of absorptive and secretory. Absorptive and secretory function. And the capsular or the satellite cells are the one which surround sensory and autonomic ganglia. So they give supportive function to the ganglia. And the next one is the Schwann cells. Schwann cells you have heard before. They are found in the peripheral nervous system and they are the special neuroglial cell type which form the myelin sheath of the peripheral nervous system. So the Schwann cells are only found in the peripheral nervous system. The astrocytes, these are the star-shaped cells which has numerous processes. They are the special cell type found in the blood-brain barrier. They help to form the blood-brain barrier and also give the mechanical and metabolic support to the surrounding neurons. The next one is the oligodendrocytes. Oligodendrocytes are the ones found in the CNS, and they form the myelin sheath in the central nervous system. So this is a place where you can clearly remember the Schwann cells form the myelin sheath in the peripheral nervous system, and the oligodendrocytes form the myelin sheath in the central nervous system. Also, they give the supportive function to the central nervous system neurons. So these are the prominent type of the glia in the white matter. And then we have the glioblasts. These glioblasts are the mother cell types from which the macroglial cells differentiate. So they are found deep in the ependyma, and they proliferate into mature macroglial cells. Okay. And then you have these microglia. Microglia are the small glial cells. They have a flat cell body with short few processes. They have defense and immunological function in the nervous system. Okay, they are the cells which give the defensive function and the immunological support for the nervous cells in the nervous system. And there are some other supporting cells which help to ensheath the motor and sensory nerve endings. In this picture, you can see the pictures of various neuroglial cells. In the peripheral nervous system, you find satellite cells and the Schwann cells. In the central nervous system, you find the ependymal cells, oligodendrocytes, astrocytes, and the microglia. So, we have talked about the special glial cells which form the myelin sheath of the nervous cells. So, according to that, whether there is a myelin sheath or not, we can divide the nerve fibers into myelinated nerves and the non-myelinated nerves. So, this table shows you some differences between the myelinated nerves and the non-myelinated nerves. Myelinated nerves have the myelin sheath. So, the myelinated nerve fibers are enveloped by myelin sheath. Non-myelinated nerves, there are no myelin sheath. Myelinated nerves have variable numbers of concentric layers of Schwann cell plasma membrane. Therefore, they are large fibers. But in the non-myelinated nerves, they are simply enveloped by the cytoplasm of Schwann cell. That is called the neurolemma. Therefore, they are very small fibers compared to the myelinated nerves. In myelinated nerves, there are Ranvier nodes in between the Schwann cell layers. So, they are the constrictions in the myelin sheath due to interruptions at regular levels. In the non-myelinated fibers, there are no Ranvier nodes. You can see here the Schwann cells form myelin sheath, and in between there are constrictions. Those are the nodes of Ranvier. The number of nodes of Ranvier are important in propagating nerve signals. Okay. Then there are these myelinated nerves. Their myelin sheath is formed by the Schwann cell in the PNS and the oligodendrocytes in the central nervous system. They give structural and metabolic support to the nervous cells and also act as an insulator. Then let's go to the sensory receptors. Sensory receptors are the nerve endings or specialized cells which convert stimuli from the external or internal environments into afferent nerve impulses. So, these receptors gather signals from the outside world and they convert those impulses into afferent nerve impulses to be transmitted to the brain or the spinal cord. These impulses, the impulses pass into the central nervous system where they initiate appropriate voluntary or involuntary responses. So, the central nervous system responds according to the impulses getting from these sensory receptors. In morphological way, we can categorize these sensory receptors into simple receptors and compound receptors. Simple receptors are the free, branched or unbranched nerve endings, free nerve endings. They are rarely visible in the light microscope, and these are the receptors responsible for temperature and cutaneous pain. And the other type is the compound, very organized sensory receptors. They are associated, they have the organization of associated non-neural tissues. So, these compound sensory receptors can be seen in the eye, ear, and those receptors for smell and the taste. In a functional way, we can divide sensory receptors into exteroceptors, proprioceptors, and interoceptors. Exteroceptors are the one respond to the stimuli from the outside of the body. They get the stimuli and the signals from the outside of the body. Those are the examples: touch, light pressure, deep pressure, cutaneous pain, temperature, smell, taste, sight, hearing, and those things. Proprioceptors are the one located within the skeletal system. So, they provide the conscious and unconscious information about orientation, tension, and the movement of the skeletal system and position of the skeletal system. So, the examples for proprioceptors are the vestibular apparatus of the ear, tendon stretch receptors, and the neuromuscular spindles. They give information on the orientation and tension, movement, and the skeletal position. The interoceptors are the ones response to the stimuli from the viscera, internal organs. Examples for the interoceptors are the chemoreceptors of blood, vascular or pressure baroreceptors, distension of the hollow viscera such as bowel and the bladder, visceral pain, hunger, thirst, well-being of the body, These are the things. These are the examples for interoceptors. So, there are some specialized sensory receptors we can talk individually. So, one of them are the free nerve endings. They are the simplest form of sensory receptors. They have numerous small terminal branches of afferent nerve endings. So they have slow conduction, and the nerve endings doesn't have myelin, and there are Merkel nerve endings for light touch sensation. Another one is the Meissner's corpuscles. They are small encapsulated sensory receptors. They are small encapsulated sensory receptors which are found in the epidermis of the skin. These are responsible for the light discriminatory touch. Another one is the Pacinian corpuscle. They are large encapsulated sensory receptors. Pacinian corpuscles are responsible for the pressure, coarse touch, vibration, and tension. These are found in the deeper layers of the skin, ligaments and the joint capsules, mesenteries, some serous membranes, some erogenous areas, and some viscera. They have appearance of an onion. Another thing is the Ruffini corpuscles. They are spindle-shaped and found in the soles of the foot. And this neuromuscular spindle is found in the skeletal muscles. They are the stretch receptor organs within the skeletal muscles. They regulate muscle tone via the spinal stretch reflex. They are encapsulated lymph field and fusiform in shape. Usually, the neuromuscular spindles lie parallel to the muscle fibers. They have 2 to 10 modified skeletal muscle fibers called intrafusal fibers. Okay. Another thing, specialized sensory receptor is a pros and bulbs. These are the lining of the oropharynx and the conjunctiva of the eyes. So then let's move on to ganglion. Ganglion means a discrete aggregation of neuron cell bodies located outside the CNS. Outside the central nervous system, there are some discrete aggregations of neuron cell bodies. They are called ganglions. We can see 3 types of ganglions. Those are the spinal ganglions, sympathetic ganglions, and the parasympathetic ganglions. The spinal ganglions lie on the posterior nerve roots of the spinal cord, and it contains the cell bodies of the primary sensory neurons. They are encapsulated by condensed supporting tissue. And the sympathetic ganglions are the multipolar ganglion cells in the sympathetic nervous system. Their nuclei are eccentric, and the cytoplasm contains lipofuscin granules. The parasympathetic ganglions are the ganglions located near or within the effector organs. They contain cell bodies of the terminal effector neurons. The parasympathetic ganglion can be a well-organized moderate-sized one, as in the otic ganglion, or a few clumped cell bodies forming a tiny ganglion. Then we have the synapses. Synapses are a type of intracellular junctions in between the nerve cells. So these synapses are the places responsible for transmitting signals from one nerve cell to another one. So they are highly specialized intercellular junctions. They allow the communication by linking each nerve's pathway. When we talk about a given synapse, the impulse conduction within the synapse is unidirectional, always unidirectional, but the response which occur for that impulse may be either excitatory or inhibitory. So synapse has a presynaptic and postsynaptic membranes. Presynaptic membranes we can see in the efferent neuron. No, sorry, presynaptic is in the efferent neuron. And the postsynaptic is in the efferent neuron. In between there is a synaptic cleft. This is a picture of a synapse. This is the first neuron. This is the synaptic end bulb. So this is the presynaptic membrane. Near this presynaptic membrane, there are synaptic vesicles containing neurotransmitters. And the synaptic end bulb is dense with mitochondria. This is the postsynaptic dendrite, the nerve which received the signal from the first one. So it has receptor ion channels for the neurotransmitters released from the presynaptic membrane. In between these presynaptic and the postsynaptic membranes, we can see the synaptic cleft, to which these neurotransmitters are released. So there are three synapses types according to the structures connected by the synapse. First one is the exodendritic type. It is the most common type. In there, axon of one neuron synapse with the dendrite of another neuron. Exosomatic one is the one where a axon from one neuron synapse with the cell body of the another neuron. Exoaxonic one is the synapse where a one synapse from one neuron synapse with one axon from one neuron synapse with the axon of another neuron. So then there is the one, the motor end plate. Motor end plate or the neuromuscular junction. This is the special synapse, type of synapse which formed in between a neuron and a skeletal muscle fiber. So, motor end plate or neuromuscular junction is a highly specialized intercellular junction which allow communication between neurons and skeletal muscle fibers. So, the neurotransmitter of somatic neuromuscular junctions is acetylcholine. This neurotransmitter acetylcholine is deactivated by acetylcholinesterase between successive nerve impulses. This acetylcholine neurotransmitter need to be deactivated in between successive nerve impulses. Otherwise, the nerve fiber will continuously kept stimulating. So this picture I don't know whether you can clearly see, is a picture of a motor end plate. This is this one is the nerve cell and this one is the muscle fiber. There are some special features in the motor end plate which is different from the normal synapse, regular synapse. Those are we can see that one motor neuron can innervate more than one muscle fiber. One motor neuron can innervate more than one muscle fiber. Motor neuron and the muscle fibers which it supplies together constitute a motor unit. One motor neuron and the muscle fibers it supplies all together constitute a motor unit. To supply more than one muscle fiber, the terminal part of the axon is divided into several branches. Each branch terminate as a motor end plate on a different skeletal muscle fiber. So one motor neuron's axon divided into several terminal branches, and each terminal branch act as a motor end plate on different skeletal muscle fiber. Altogether, this motor neuron and the skeletal muscle fibers it supplies constitute a motor unit. So the muscle surface occupied by the motor end plate is called as the sole plate. And the postsynaptic membrane form secondary synaptic cleft. That means when the postsynaptic membrane of the muscle fiber is forming cleft and it is irregular in shape. Okay, irregular in shape like this. And therefore, the irregular synaptic membrane forms a secondary synaptic cleft. And the overlying presynaptic membrane is also irregular. Both the presynaptic membrane and the postsynaptic membrane are irregular. Because of that irregularity, there is a primary synaptic cleft and secondary synaptic cleft. These are the special features of motor end plate. So you have to remember these things because you may get questions on listing differences or compare and contrast regular synapse and motor end plate like things. Okay. Then I have included a small note on blood-brain barrier because this is seen only in the nervous system. This is consists of structures situated at the capillary level near the brain. Blood-brain barrier allows a selective entrance of blood contents to the nervous system. So unlike in the other parts of the body, the capillaries in the brain can't diffuse or actively pass everything in the blood into the brain. This blood-brain barrier acts as a gate, which select things from the blood and only allows those selected things into the nervous system. Therefore, blood-brain barrier prevent toxic and harmful substances to reach the brain. So the blood-brain barrier comprises of the capillary endothelium without openings. This is the capillary endothelial cell. They have very tight junctions in between the cells, so there are no openings. And then the basement membrane. Then the basement membrane of the endothelium. And the terminal processes of astrocytes covering the capillaries. These capillaries covered by the terminal processes of the astrocytes. Then the intercellular space containing fluid and the process of the cell bodies of the neuron. So, between the neurons and the blood, there are several layers which only allows selective particles from the blood to enter the nervous system. That's why certain drugs and certain toxic substances can't reach the brain under the normal circumstances. Okay. So, that is the all in the nervous system lecture. You can use this lecture as a guide for the nervous system histology part and also a complete lecture note. I will upload this to your WhatsApp group because I don't know whether the Moodle is working these days or not. Then you can get the PowerPoint presentation and please refer the Vita's histology textbook for further references. And you will be having a histology practical on nervous tissue. I think it's on today. So go through that practical and the lecture together. Okay. And do you have any questions? I can't hear anyone is talking. Are there any questions? I think according to the silence you are giving, there will be no questions. So thank you for listening this boring lecture. Okay, good luck and have a nice day.
Thank you, ma'am.
Thank you, madam.
Thank you.
Thank you, madam.
Thank you, madam.
Thank you, madam.
Thank you, ma'am.
Thank you, ma'am.
Thank you, madam.
Thank you, madam.